See if there's a word. Shall we go ahead and get started? All right. I went ahead and clicked record, so.
Oh, you already did?
So, yeah, all right. Hi, thank you for coming to the Science Bubble tonight, and thanks for folks who are listening. We're here to talk about science tonight. This is a partnership the library has with Cal that we're really excited about, and we encourage you to come back and check out our monthly program. I'm going to do a quick land acknowledgement, and I'll introduce the program, and then our team will introduce tonight's speakers. So, the Berkeley Public Library buildings are built on the territory of the Hyunchul within the land of the Chochenyo-speaking Ohlone people. We acknowledge and honor the Ohlone people who have lived in Berkeley and throughout the East Bay time since immemorial. The library recognizes that Berkeley residents have and continue to benefit from the use and occupation of unceded stolen land, and we pledge to recognize the permanence and the rich contributions that the Ohlone people continue to bring to the city of Berkeley and throughout the East Bay. We celebrate the strength of the Ohlone people and their culture. The Berkeley Public Library is committed to helping create a more just and anti-racist society and to furthering the visibility and celebration of Ohlone and authors who are specific to the East Bay and Ohlone culture in our collections and programming. All right, so welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and to create constructive discussion about a variety of science topics. We have two speakers at each seminar who will talk about their current research or a topic that they find really interesting. The organizers are three graduate students at UC Berkeley, Helen Zong, Daniel Bretthauer, and Madison. Lectures happen on the third Tuesday of the month at 5:30 p.m. These programs are live streamed on the Science Bubble YouTube channel, so you can view them later and share them with others. Popping the Science Bubble also has a website and a listserv that you can join to keep up to date on new Science Bubble lecture topics. All right, enjoy.
Hi, everyone. Thank you for being here. We're excited to have you tonight. As Kelsey introduced us, we're Popping the Science Bubble, bringing scientists from the UC Berkeley campus to community talks here at the library. We're here third Tuesday of every month, and at the end of the talks, we will throw up socials and QR codes in case you want to join our listserv in order to hear about future events. But thank you for joining us tonight. And I'll introduce our first speaker, Hyunchul. So Hyunchul was born in Pohang, a city renowned for its leading scientific community in South Korea. His early fascination with science led him to pursue a career in chemistry. He then earned his undergraduate degree in chemistry from Pohang University of Science and Technology and is currently pursuing his PhD in inorganic chemistry here at UC Berkeley. He works as a researcher specializing in single molecule magnets. Outside of the lab, he enjoys cooking, binge watching shows, and playing badminton with his wife. He's passionate about both his work and sharing scientific knowledge. And we're excited to have him here today. So please take it away.
Thank you for the nice introduction. Yeah, as it is introduced, I'm Kwon. I'm a PhD candidate in UC Berkeley Chemistry Department. And today I want to talk about the presentation with the title of Single Molecule Magnet, Chemic Blueprint for Future Data. But before I start, I want to ask to the audience that if you have ever heard about single molecule magnet before?
No.
Oh, no. You have heard of it?
I was just reading about them in that book.
Oh, yeah, this is, I think it's a good book for magnet, but today we are talking about single molecule.
She mentions them towards the end of it. Yeah.
Thank you for sharing that. Yeah, so before I came here, I asked ChatGPT about what is the single molecule magnet, and ChatGPT gave this image. So this is obviously not a singular image, which means it's good because you are gonna learn new information that ChatGPT doesn't know. Yeah, so let me start.
Why is that not a single molecule?
So I, as a chemist, I don't think this is molecule at all.
You don't think what?
This is a molecule at all. This
It's
is
not a
some-
molecule.
Oh,
Yeah,
okay.
something, I don't know, bioorganism? I don't know. Yeah, so, it's yeah, let me start. So 96 million messages sent, 1.5 billion swipes in the Bumble, or 500 hours of content uploaded on YouTube. These are what is happening every minute in real time. So every minute on the internet, we create a need for the data. So here, what I'm showing is the plot that y-axis is the year and x-axis is the size of the data. You can see that the exponential increase of the annual size of the global data field. So people are trying to find a way to store this data, and like we are expecting that by 2025, we're gonna have around 200 zettabyte of the data. Zettabyte here is 1 zettabyte is 10 to the 21. So some people call that there will be a zettabyte apocalypse, which means that there are too many data compared with the size of the data storage device or number of data storage device. So we cannot really store all the data that we put. This is what they call zettabyte. So to prevent this kind of apocalypse, we are trying to develop the good data storage units, data storage device that can store the data in a compact, more dense way. The current technology, we are using this magnetic hard disk drives, which we call HDDs. And in these hard disk drives, this is what we are using in the computer or smartphone or everywhere. And the way it works is inside this magnetic hard disk drive, there are other small magnetic domains, which is made of the magnetite or cobalt or any kind of magnetic metal, which the size is around 200 nanometers there. So here you can see that in this picture, there are several domains you can see, and their colors are different. Some are gray, some are white. Those are showing the data is whether it is 1 or 0. So each domains are the magnet, which the size is around 200 nanometers. Scale. Yeah. So as I told you, the size of data increase, people are trying to develop the device that can store more data in a more dense way. And you can see that the data storage device has advanced as time goes. So we were expected to reach the data storage performance of 1 nanometer square per bit as an expectation. However, there is a lot of physical limit that couldn't make this happen. So in reality, this rate of development is slowed down. So as a chemist, one could think about what if we store the information in a single molecule? So use the single molecule as a data storage. And here is the single molecule that is first shown to be showing. This is the first single molecule that is going to store the data in here, which is behaving as the same way as the magnet. And here's the pop sheet. I prepared this for you. So can you guess how large this molecule would be?
And microscopic, I mean,
Yeah, this one single molecule. How large it is?
well,
We can't see it.
you can't see it.
Yeah, you cannot see it, but if you use the unit like meter, nanometer, micrometer.
1,000 nanometers squared.
No, I mean, I don't know what the division is. If you take, what the tiniest is, but it's like way small. I mean, the only thing smaller is an atom.
Yeah.
Yeah.
Yeah, it is. So yeah, in the previous slide, I show you the current state of art technology of storing memory. One unit is like 200 nanometer scale. And this molecule, the size is about 16 or 17 Armstrong. And 10 Armstrong is 1 nanometer. So the size of this molecule is around 2 nanometer, which means in 4 nanometers square area, you can store A information or A bit. So this is really small. Like, cannot imagine the smaller thing than this one, but yeah, you're right. Single atom, this is a collection of atoms, and single atom is usually mostly like a 1 Armstrong, and we have like these, let's say, 20. The size of this molecule is like 20 atoms placed in a row, right?
Is it smaller or larger than a tardigrade?
What is that? Pardon?
A tardigrade. A tardigrade is a water bear. It's not able to be seen either.
Yeah, water bear. I think it's much
Smaller than a tardigrade.
like. Let me do bigness. I think like we need the 1 to the 8 of this molecule to make the comparable size of the micro. And tardigrade is, I think, the micro.
It is.
So yeah, so in this very small molecule, we can store the data. Yeah.
Maybe over there, early, but what do you mean by having this one?
Yeah, that's exactly what I was gonna talk and spend most of the time, most of the seminar. Yeah. Give me 2 minutes.
No, I'm sorry.
So if we can use single molecule as a data storage unit, then we can reach to the point where we can store the 1 bit in a 1 nanometer scale. And this is the motivation of why we want to use molecule to store the data. Then, so as I told you earlier, that we are storing data in the magnet. But before I go into how actually single molecule magnet store the data, I want to explain how magnet works or how magnet store the data. So here I'm showing you a cup, which is made of ceramics, and the magnet. Usually it's a magnetite, or I'm showing the formula here. So for the cup, when you apply the magnetic field, you can see the, here, this is, so I'm a PhD student, so I need to explain physical phenomena by plot. And here in this plot, the x-axis is the applied magnetic field, and y-axis is the magnetization. And when you're thinking about you're applying the magnetic field to the cup or ceramics, then you see the response as the magnetic field increase. And then when you remove the magnetic field, then the magnetization value goes to 0, which means magnetic field doesn't do anything permanently once the field is removed.
Once the what?
Once the magnetic field is
Okay,
removed, then there's nothing changed for the curve.
the data is still on there.
No, curve is not, we cannot use curve for data.
Oh, okay.
So this is like, I'm trying to explain why magnet is special and why in magnet we can use magnet to
Store data.
Yeah. And let's see how magnet would work under applied magnetic field. So here, in magnet, magnet tends to disappear for the cup. For the magnet, when you apply the magnetic field, the magnetization increase. And then when you remove the magnetic field, then you can see that magnetization still persists under zero applied field. Which means after you apply the magnetic field and remove the field, there's something changed and magnet becomes magnetized. This is exactly how people generate magnet, like bar magnet or the magnet you use to attach to your refrigerator. And so under zero field, this magnetization persists. And so I'm just showing the magnet field versus magnetization plot of this magnet. And you can see that under zero field, actually, let me explain how this plot is made. So you apply the magnetic field, then the magnet is magnetized. And then when you go to zero field, this magnetization persists. And then you apply the magnetic field to the opposite direction, which is this way. Then you magnetize in the other direction. And then you go to zero. Then magnet remains magnetized on the opposite direction. So this means that under zero field, I should have mentioned, I should have put here zero. Under zero field, there are two possible states that magnet can exist, which here we call is 1 and 0 in terms of data storage application. So this is how magnet works in hard disk drive. And single molecule magnet behaves in almost the same way as the bulk magnet. Single molecule magnet chemist says the typical magnet as a bulk magnet. So single molecule magnet is a molecule that shows the property similar to the bulk magnet. As you can see here, on the zero field, there is a spin up and spin down in the zero field, which we can say is 1 and 0. Yeah, so this is why we can use the molecule to store the data, since on the zero field there are two possible states of the molecule. But the problem of single molecule magnet, and the reason why we don't use single molecule magnet in everyday life, is that this kind of magnetic hysteresis behavior already persists up to very low temperature. So this data is measured at 2.2 Kelvin. So 0 Kelvin is absolute zero, and 173 Kelvin is the 273 Kelvin is the temperature where the ice freezes, and this plot is measured at 2.2 Kelvin. So this is extremely low temperature. And as temperature goes higher, this kind of magnetic hysteresis behavior or magnetic hysteresis loop is closed. And now, at certain temperature, this molecule is no longer behaves as a magnet, and this is rather behaves similar to the cup. That's why we cannot really use the molecule yet for the data storage. But as I mentioned, this is the first single molecule magnet that is reported like 30 years ago. And I want to show you the state of art single molecule magnet that our group has made at UC Berkeley. So 30 years ago, this first single molecule magnet was made. This is 2 nanometer wide, and it only shows the magnet property under 4 Kelvin. And now, like 2 years ago, we have published this nice molecule where it shows a magnetic property or magnetic hysteresis up to 72 Kelvin. And the size of this molecule is much, much smaller. It's only 0.8 nanometer. So we've been making an improvement. And one achievable target is the 77 Kelvin, where liquid nitrogen, nitrogen is liquefied, and we can, it's the temperature where we can keep in a reasonable point. So we are approaching to 77 Kelvin, and yeah. And I think probably you're interested in how we are actually making this molecule. So in the laboratory, we are making molecules in a largely as 3 steps, not studying molecules largely in a 3 steps. First is the synthesis. So we synthesize the molecule and then isolate the molecule in a different forms. This can be a powder or single crystal. And then once those are isolated, using the spectroscopic technique, we characterize the magnetic property or structural parameters of the molecule. Yeah, those are how the research is conducting in the laboratory every day. Yeah, so this is the final slide. So I introduced you what is the single molecule magnet and how it works. And here are the current limitations for the practical application of this single molecule. First is the operating temperature. So the current record of the molecule, the temperature that the molecule operates is only 72 Kelvin. As I mentioned, for practical application, this temperature has to increase up to at least 77 Kelvin, where liquid nitrogen, where we can use liquid nitrogen to keep that temperature. The second thing is the reading and writing mechanism of this reading and writing mechanism of magnetization inside this molecule, because since the molecule is so small, we have yet developed the technology of how we would actually read and write on the molecule. So that is the other research topic that people are pursuing. And third thing is the chemical stability. So most of the molecules that I've shown you here are pretty much air sensitive or thermally unstable, but there are many problems. So if we want to put those molecules in the smartphone and use it anywhere, then those molecules have to be very stable. So that's the third thing that we need to tackle for the future. Yeah, so this is end of my talk. As you can see, this is very collaborative, multidisciplinary international research. And I thank for national science funding and UC Berkeley that enables me to work on this. Thank you. Do you have any? Oh, actually,
I'm sorry, you had the word hysteresis.
yeah, I should have.
Can you tell me what that means?
So before this magnetic hysteresis, let me. So magnetic hysteresis is that when you magnetize the sample, and then you apply the field to the other direction, the magnetization doesn't go to zero, but goes to stay at certain value. And if you want to remove this magnetization, you need to apply certain amount of magnetic field to the opposite direction. So hysteresis is basically the magnetization of the molecule. The better way to explain is that before this hysteresis is a loop, and here the loop is open in the plot, which means the magnetization is not exactly following where you apply to the value where you apply it. Maybe I'm confused, but is it answered?
I didn't mean that to be a trick question.
Yeah, I think I should.
Thank you.
I'd love to know a bit more about how you and your lab have decided to test different molecules. Like I assume that you're not just like, yeah, let's throw together a whole bunch of metals and see what happens. Like how you decide what chemical you're going to synthesize.
Actually, yeah, we just throw everything. No, not that one. Yeah, so there are a lot of modeling by actual drawing. So you first need to consider how the electron would behave inside the molecule. So let's say for lanthanide, so basically the original magnetism or magnetic field is from unpaired electron, and how those unpaired electrons are occupied in the molecule, in the orbital. So we first was to design that, and to design that first we want to choose what metal we're going to use, and second what ligand field. Ligand field is not a good term for this field, but ligand field we want to apply, something like that, and then we can ask for a computation collaborator to calculate the electronic structure of the molecule. Then we can move on to synthesis. Or we can just synthesize it first and measure it. And if the measurement is interesting, then we proceed on other stuff further. Yeah. I can talk you more detail. Yeah.
Is there a relationship between the size of the molecule and the temperature that they're stable at?
That's a very good question. So long ago, when the first single molecule magnet is out, people have been thinking that if the molecule size is bigger, the blocking temperature or the temperature where it behaves as a magnet will go to higher, but that was not true, because there is so in designing single molecule magnet, we first want to consider how many spin center or metal atom we have. But at the same time, we want to consider how anisotropic, magnetically anisotropic it is. So you can imagine that there's no Bohr magnet, right? In the Bohr magnet, there's no Bohr magnet. Oh wait, there's a Bohr magnet, and it's not a thing. Anyway, the magnet has to be somehow anisotropic to store the information, and or to behave as a magnet. So I'm thinking if the magnetic anisotropy is consistent, then the bigger the size, the temperature will higher. But controlling both the number of matter atoms and the magnetic anisotropy is the challenge. Yeah.
So obviously, Barman has are many molecules that are not hundreds. Right, you're saying the problem is that like, you wanna make sure that the different charges stay very low to them. And like, don't get too dispersed. So like, like, I guess, just the follow-up question is like, has anyone looked into rather than single molecule magnets, like doing 4 molecules of the same that can maybe pack together? Or like, is there work on that scale? Does that make that like, instead of being hundreds of molecules that make a bar magnet, or a single molecule is just like a small difference?
Yeah, that's perfect question. I think that's two good questions. So I think I need to think about it. But in general, like when you're talking about when you define the molecule, then we assume that in the molecule, actually we are looking at the spins, not the atoms. So in one molecule, there's a one electronic state inside the molecule. That's why we are thinking it's a one molecule. And if we simply pack several molecules, I think those electronic states are not communicating. So they will create in a as a separate molecule. But if we can think about a way to couple them, that would be an interesting topic. As far as I know, like if they are coupled, then we consider that as a one molecule. Like 4 molecules becomes a one molecule again. So yeah.
So then it's just a bigger molecule.
Yeah, it's a bigger molecule.
If we don't have any more questions, I think that's it.
Thank you so much.
Thank you, Hyunchul, for the really interesting talk. While our next speaker is setting up, I'll introduce her. So our next speaker's name is Elleman. She grew up in Rhode Island before coming to Cal for a bachelor's degree in chemical biology. She loved the Bay so much that she decided to stay and complete a PhD in chemistry at Stanford in 2021. And then she returned to Cal for her postdoc in molecular and cellular biology. In her free time, she enjoys skiing, hiking, travel, and all food and food-related things. So take it away.
Just to confirm, right now I have the screen muted because sound's coming over there, is that correct? Okay, awesome. So today, I'm going to be telling you all a bit about some work I did during the PhD, trying to figure out how to control electrical currents to and from the brain and within the brain itself. And so I did that PhD at Stanford University in the Dubois Lab in the chemistry department, and I've since moved on to doing a postdoc at UC Berkeley in the molecular and cellular biology department in the Kramer and Brophon labs. As I'm talking, definitely ask any questions you'd like to ask. Stop me if I'm being confusing or if I'm not clear. I definitely trip over my words. So yeah, the long and short of it, start going here, is that you are comprised of many cells, billions and billions of them, which all need to communicate to each other. And I'm going to tell you a little bit about how a subclass of cells called neurons talk to each other, and how we're looking into controlling how they talk to figure out how different pieces of you connect. How like what you sense in your toe, how that makes all the way up to your head, how your brain then tells different parts of your body what to do.
So the main system I'm going to focus on here is called the nervous system. So this is comprised of the central nervous system, so that's your brain and spinal cord, and then your peripheral nervous system. So that's all the nerves in the rest of your body which send information to your brain and receive instructions back from your brain. The actual items that transmit these instructions are things called neurons. So these are specialized cells that transmit information to and from the brain, up and down the path of the cell. So for example, I've oriented this guy here so that the input will be coming somewhere down like near your toe. The signal gets transmitted all the way up the neuron, and then the output would be all the way up into your spinal cord, which is to say that these neurons are responsible for transmitting sort of any information you can think of your body needing to communicate within itself. Things like thoughts, memories, senses, motion, like telling your arm to move, the neuron is transmitting that signal. And these things can be either really, really small, like the neurons in your brain, so a few millimeters long, or absolutely huge, like the one I was just describing. So going all the way from your toe, all the way up to your spinal cord. That can be one set transmitting that signal. So in order to do that, your body and your cells need to be able to transmit this information really, really fast. So at up to 250 miles per hour. And that allows you to instantly respond to things that you sense happening, or to transmit your thoughts very rapidly to the rest of your body to tell it what to do. And the way it does this functionally is actually just electric current. In the same way that we build wires to transmit information to tell a computer what to do, or to tell some motor what to do, your body uses cells to transmit that same type of information. Yeah.
So, for 1 meter long neuron, or neuron in general, where are they? What kind of tissue are they located in our
Yeah,
body?
so your neurons in your spinal cord go in through your vertebra, basically. That those are designed to protect that sort of central tract of neurons. Your brain has a skull around it to protect that sort of central location of neurons. In your peripheral nervous system, because you need to sense things across your skin, you need to sense your muscle state. You have neurons in the peripheral nervous system that innervate different points all the way around your body, and they connect to other cells that might individually be responsible for sensing information, like your skin cells or muscle cells. But you will have neurons sort of like fingers innervating those tissues to transmit that information all the way back up to your brain.
So are they like kind of intertwined with the blood vessels, or?
It really depends. I'm not an expert in medicine at all, but I know that human body diagram where they show all of your vascular system, there is a similar nervous system diagram, and it does sort of overlay in different places. The tricky part is modern medicine, modern science doesn't actually have the best sense of where all the different nerve tracks are. That's one of the things that makes surgery so difficult, and treating people after surgery is we sort of know how to sew your skin, how to sew muscle tissue, but nobody really guarantees that the neurons get put back in the right place. And that's one of the reasons a project like this is important, is trying to figure out how everything connects, because they're so thin and so, I guess, spread out that they innervate everywhere, that you could wind up with like nerve damage and things post-injury because they aren't properly repaired, just because we don't really understand fully how they interconnect. All right. So as I mentioned, the way your body is transmitting information is most basically through electric current, but it does it a little differently than how you usually think of wires working. We're going to talk about how that is. So when you think about current through a wire, for example, using a battery to light up a light bulb, what we're actually talking about is the flow of ions from some wire through the positive terminal of the battery. Negative terminal spits out ions, and sort of round and round you go, and that flow of ions, that current, is what gives you the energy to power something like a light bulb. In your cells, it's a little bit different. That rather than the current flowing down a wire, it's actually flowing across your cell membrane. So each cell in your body is going to have sort of like a balloon-like exterior, a membrane to it. It's that encase a bunch of ions in water. And outside of that cell is going to be more water with a whole other bunch of ions in it. And that membrane is going to be basically impermeable to those ions. So much as oil and water don't mix, you can think of the lipid bilayer as being kind of an oil bubble around a water droplet, and it's not going to let the water or those ions through. So in order to conduct current across a cell, what we need are channels to let those ions through. And ideally, to control information flow, we need channels that only allow specific ions through. So these channels are usually proteins that sort of act as like cylinders to let those specific ions through. And often they sort of select for different ions based on size or charge or some other property that allows them to differentiate what specific ion is going to go through the channel. This eventually allows current flow. So for example, the movement of positive ions into a cell, which produces, a voltage potential across that impermeable membrane, so that you have a driving force for ion movement. And this sort of potential energy across that membrane becomes the driving force for information, that flow of current that goes from cell to cell to cell and transmit all the information that needs to be transmitted between your periphery and your brain. So get into this a little bit deeper. If we look at that neuron I showed on the front page, in order to transmit information super rapidly, so 250 miles per hour, you might imagine it's kind of hard. You have a meter-long neuron to keep that current flowing back and forth across the membrane all the way up that huge neuron. So your body actually has a good way to make communication even faster than what that would naturally be. And that's by skipping bits of the neuron. It basically, it wraps around extra cells, so extra lipid membranes, around portions of the neuron so that they're more insulated. And this is called a myelin sheath. This would be myelinated neuron here, for example. And that prevents any kind of channels from localizing, any kind of current from flowing over those insulated bits of the cell, which effectively allows the signal to then jump and skip certain lengths of the neuron, which makes the actual transmission of the signal going into the cell, down the membrane itself, out of the cell to the next neuron. It makes it much faster. And so if we zoom in then at these little areas, these nodes, which are actually doing the conduction of current across the membrane, they're actually responsible for signal transmission. We have two important types of channels. The first are, just to summarize, electric signal is jumping between these nodes and it's enabling that very fast signal transduction. Without these nodes, we'd be talking about maybe 1 mile per hour signal transduction. With them, again, you get up to 250 miles per hour, so much faster. The nodes themselves are conducting current through two types of channels, both of which let positive ions across. The first are sodium channels. These allow sodium into that node in response to like a teeny little signal. So like, let's say you feel a hair brush your leg. That'll be enough to open those channels so that you have this influx of positive current. And that becomes this sort of upswing here of something we call the action potential. So the physical electrical signal is being sent down your neuron. After the sodium channels open, they close. The potassium channels then open and they let potassium out of that node. And this becomes the downswing of your action potential. So together, what you're creating is this spike of current that is then sent from node to node to node down your neuron from cell to cell. And it's allowing you to transmit information very rapidly according to the size and pattern of these signals that are played through these nodes. Does that make sense so far?
So the rate of the signal is decided by how fast these ions are penetrating.
It basically is determined by what type and how many of these channels open at these nodes. So if you have a lot of sodium channels open, but there's not many potassium channels open, you're going to have a really tall spike. If you have things open and closed, both of them very rapidly, you'll have many spikes in a row. And you can basically modify how the signal looks and the pattern is played to transmit specific types of information.
So what is the time scale of
So an action
ion
potential.
moving and the channel opening?
So ions diffuse, I think on something it's like order of 1 per 10 to the 9 seconds. It's really fast. Action potentials themselves take a couple milliseconds. So it's still very rapid. All right. So just to summarize, the action potential is the electric pulse or the current flow that's carrying information from cell to cell to cell down your neurons. So if we want to start looking at how our neurons talking to each other, what we actually need to figure out is how we take this signal, which is traveling down our neuron, jumping from node to node to node really rapidly. How do we stop that signal? How do we control which neurons are talking to each other? And the way we tackled this during my PhD was we decided that we were going to target the voltage-gated sodium channel. So the channel that's involved in starting that action. We decided we're going to figure out a way to block it very precisely, so now we can control which neurons are talking to each other. To do this, we actually relied on a molecule that nature produces. So there's this small molecule toxin called saxitoxin, which is produced by algae and cyanobacteria. And where you might have heard of it is if you've ever had somebody get relative shellfish poisoning, this is the toxin that does it. It's really, really potent. It binds sodium channels really, really tightly, which means that for you, if you were to like eat it, it's very, very important because it stops all of your neurons in your body from transmitting any signals to each other. So we don't want something that's quite that good, right? We only want to stop neurons from talking in specific places and when we want it to. We don't want to just kill everything we put it on. We have to work on modifying this small molecule inhibitor. The way we thought about doing this is so functionally this small molecule itself, this is just a colorized picture of our channel here, and then rotated to show basically the hole through the channel on the right here. Functionally, the small molecule acts as a cork. It just physically blocks the pathway that sodium ions would go through. But again, we don't want it to block all the time. We basically want to modify it so that sometimes it can't block. And then when we want it to, it corks things up. You have no more ion flow, you have no more signal, you stop that neuron from talking. The way we decided to do this was by sticking something called a photopage on saxitoxin. So the photopage piece is everything on the right side of this molecule. Now this is basically a stick diagram showing a bunch of atoms lined up together, bonded together. The molecule is comprised of carbons and nitrogens and oxygens. Functionally, that doesn't super matter. The important part is that by virtue of sticking this photopage on, when we add light to this molecule, we enable chemical bond cleavage. So specifically, the cleavage of this bond here, which I've marked out in red, between the inhibitor and the photopage. So what this allows us to do is basically take something big and bulky that absorbs light, stick it on our inhibitor to make it inert. That's now too big to cork up that sodium channel. But when we hit it with light, we release the free inhibitor, and now it's going to block everything again. So hopefully then what this allows us to do is basically turn off neurons with light wherever we want to. If all you do is feed a cell, an animal feeds you this molecule, it does absolutely nothing. You hit somewhere with light, suddenly that neuron is off. The advantages of this kind of approach are that it's really high resolution, so you can basically target very, very tiny areas of a cell or of a tissue. It's really fast, so that breaking of that bond to release the inhibitor occurs over milliseconds or seconds. It doesn't require modifying an animal at all. You can just feed them, basically, this molecule. And it can be applied to live animals because it's inert before you add the light. Nothing is going to happen. If we go back to thinking about our action potential, what this actually accomplishes is, so if I look at a single neuron in a dish and I apply a little bit of some stimulating current just to make it start firing action potentials. So this here is just it firing 3 of the action potentials I pictured like this earlier. That neuron's happily trying to communicate with its friends, right? If I then apply light to that neuron, it does absolutely nothing. The neuron doesn't mind if the lights are on or off. Living its best life, it's still talking. If I apply the photopage toxin, so with that big bulky group preventing it from binding the sodium channel, again, it does absolutely nothing. It's unable to bind the channel. The neuron is still talking. But if I hit that neuron, then with light, now we're breaking off saxitoxin itself. It binds the channel, and we halt all signal transmission. The nice thing being that if we then wash off that free toxin, or if we just give it some time to diffuse away, that signal comes right back. This is a fully reversible approach to prevent one neuron from talking to any of the others. Now, the kind of cool part with this is like it's all well and good that it works with a single cell, but as I mentioned, what we're interested in is seeing how neurons communicate in like real animals in real life. We had collaborators at the Ehrlich lab in Wisconsin who basically took our small molecule and applied it to zebrafish. So zebrafish are like tiny 2-centimeter fish. The nice thing about them being that their larvae, the babies, are transparent. So it means when you apply light to them, it goes absolutely everywhere. So these guys are hanging out in the dish, and we stick a little light over them to basically light up certain neurons on that zebrafish. And we have the zebrafish bathed in our photophage saxitoxin. It's just hanging out in the water, or we can inject it into the fish itself, and it hangs out the fish. The fish is totally happy, doesn't mind it at all. When we apply light, though, to a specific area of the brain, so in this case, the brain stem, which is responsible for movement, that's going to uncage the photophage saxitoxin that the zebrafish is bathed in. And we go from a zebrafish that is wiggling around, moving its tail. So that's what these dots are here. They're just tracking tail movement. You can see it's going back and forth. It goes from having a full range of motion to when we uncage our toxin, absolutely silent. We basically halted any signal transmission from the brain to its tail. It is no longer able to move. And this is repeatable across many times of uncaging. So if you have a zebrafish where this axis has been flipped, so this is the tail moving right, this is the tail moving left, you can see it's going again, wiggling around. You hit it with light, stops moving, give it 5 minutes, motion comes back, hit it with light again, stops moving. And so what we can basically do is confirm the communication between the brainstem and the rest of the body and use that to control movement. The sort of extra fun part of this, and I'll show a video, is that because we can control where we release the toxin with where we apply light, it enables actually focal uncaging. So that means uncaging that's local to a specific location. So for example, if we have our zebrafish, we're stimulating, the tail waggles back and forth, swimming around. If we take our light and we only block signal on the left side, so it can no longer receive information from that side, you can see that the tail starts moving only right. And if you stimulate it thereafter, because it can no longer receive input from the left side, locomotion and swimming is entirely relocated to the right. And that again recovers with time. Basically, this is to say that like the whole point of the PhD was to design a small molecule that would be useful for controlling how neurons communicate with each other through controlling this electrical signal. And basically with the creation, I need to get past this page. Here we go. This chemical shown here, we basically accomplished that. And we were able to take a look at our neuron, which is transmitting that signal from one side to the other, and show that we can pulse it really, really precisely. So our collaborators are hoping to use this to basically disentangle how different parts of the body communicate with each other. So yeah, this work was done. We brought out at Stanford, a lot of good people, and a lot of awesome collaborators, which I didn't have her on the slide to show. And if anybody has questions, happy to answer them.
So, on the right, you use, that's possible because that fish was transparent.
Yeah, so light, we're using UV light, so that's very, very blue light, and that will penetrate through a tissue on the order of a few millimeters to a few centimeters. So if we were to do a non-zebrafish animal, so something like breast skin, it would make it through, maybe the first few layers of epidermis, maybe a little bit into the muscle, but it would be hard to get much farther than that.
Okay, the toxin and photophage, is that goes away?
Yeah, so basically the photophage piece itself is inert. It's not going to do anything. We tested it on cells. We tested it on mouse brain slides. We tested it in zebrafish. It doesn't seem to functionally do anything. Like basically the toxin, when appended to that piece, does nothing. And that photophage piece itself also seems to do nothing. It's sort of the only functional part of it all is this saxitoxin piece, which gets released by light.
So to follow up on this, does the photophage bind to any other molecules?
Not as far as we were seeing, like we saw no functional effect, but we didn't run basically a drug screen. You could pay a pharma company tens of thousands of dollars to like check your molecule against everything they know of, but we did not have that kind of cash on us.
So obviously this is helpful for trying to figure out like what sections of your brain communicate with what sections of your body. But how small of a section of your brain are you able to isolate? Right, like we know enough about like hemispheres or lobes, but how much smaller?
Yeah.
Get that spatial information.
So this particular type of photophage has to absorb a single photon for that bond to break. So it's called a one photon photophage. So one photon of light. That means that you can release the cage in about the size of a cell body's worth of area if you really want to. It gets kind of difficult because you have to design like a laser to have that narrow of a beam. There's a different type of photophage, which I also made, and it's published somewhere, which is called two photon photophage. It's got a slightly different structure for this component here. But the important part is that rather than needing one photon of light to break that bond, it has to absorb two photons at the same time. As you might imagine, that's going to occur much less often than just one photon being the bond. So it basically reduces the area uncaging even further to about 1 micrometer squared, which is basically down to like a portion of a cell. So looking at this picture here, the cage I'm talking about would probably uncage, or sorry, would probably result in the release of toxin everywhere on the screen over the whole cell. The two photon one would cause the release maybe around like just these last 3 or 4 stars here. You can do it over a much smaller area and then basically figure out which channels in which part of the cell are important for signal transmission, or how different parts of the membrane contribute to the actual signal.
So, two photons, you mean by two photons is that two photons with different energy?
Usually the same energy, often much lower energy overall because you're effectively kind of summing. So it means that when you have two photons uncaging, usually it requires closer to red light, so lower energy light, but you tend to need higher powers because you want two photons to hit at the same time.
Oh, so two photons could be the reason why you designed this, it kind of dissociate in a slower rate.
Yeah, so the idea behind two photon uncaging is that you can confine the uncaging area to be smaller because you need two photons to hit at the same time. You can think of it as basically you have to have two sort of laser beams crossing each other at a very team point. Whereas if you just shine one laser straight down, you're gonna get like a circle of area that have light. Whereas if you have two that cross each other, you have like a point where both of them hit. The other thing that's advantageous is because it's absorbing two photons of lower energy light. That means higher wavelengths, like red light. Higher wavelength lights penetrate further into tissue. So whereas the blue light we use for one photon uncaging can only make it a few layers into your epidermis, the red light can make it a couple centimeters in. So you could actually, like, for example, reach a portion of like the spinal cord or the brain, which is easier if you're dealing with like real animals and not zebrafish.
How flexible is the photophage applied to other, say, like toxins stuff like that?
Yeah, so this is basically a method that's been used a whole lot to try and selectively release a lot of different things around the body. What we did that was slightly different is this particular structure of cage. So everything from this point down was what was used most commonly as a photophage. You can imagine it's much smaller, it's not very destabilizing, and it also turns out it's not very efficient. So you really have to like fry the tissue in order to get it to come off. For whatever reason, and we are told to clear how, because we modified our cage with this piece mainly to destabilize toxin binding to the channel. Basically, we stuck on a bunch of negative charges so that our binding site, which likes positive charges, like sodium ions, no longer likes to bind to this molecule. But it turns out by like happy mistake that by adding that particular component, it makes this overall molecule much more efficient at photoencaging. It takes like way lower like powers to break this apart than it usually does for this class of molecules. Which is nice, but I have no idea why that's true.
Well, if there's not any further questions, we'll just go back to that.
Thank you to both our speakers tonight. These were excellent presentations.
Yeah, so thank you guys for joining us. We'll hear that they're excited of everyone.
Yep. And started with telegrams and go ahead and transmit
Yeah, my son.
molecular. And just, yeah, as a new stage in research, so she mentions that. Anyway.